Good afternoon and welcome to the Genomic webinar, Is Genomic Medicine Ready for AI? I'm Candace Chapman, Director of Marketing for Genomic, and I'll be your host today as we walk through a discussion on the promises and pitfalls of using AI for genome interpretation. Many of you are already familiar with Genomic, but here's just a 15-second overview for those of you who are new to us. Our speaker, Mark Kiel, started Genomic in 2014 when he faced challenges in finding the information he needed for his variant interpretation work, because it was nearly impossible to do any in-depth search of the published scientific literature with the commercially available tools. Out of that need, Genomic and the Mastermind genomic search engine was born. Now, Genomic powers evidence-based genomics for clinical labs, pharma, and life sciences with tools and services designed to break the variant interpretation bottleneck. And you can learn more at genomic.com. As you're watching, please feel free to submit questions to Dr. Kiel through the chat window on the right or on Twitter at Genomic. Because Mark will be showing examples from the Mastermind genomic search engine near the end of his talk, I invite you to follow along with him in Mastermind. If you don't already have a license, you can sign up for Mastermind Free Edition at the special link on the screen, bit.ly/AI webinar 2018. So without further ado, I'll introduce our speaker. Dr. Kiel is founder and chief science officer here at Genomic, where he oversees the company's scientific direction and product development. Prior to starting Genomic, Mark completed his residency in clinical pathology in 2014 at the University of Michigan. While at Michigan, he completed a fellowship in molecular diagnostics and devised the informatics framework for clinical next-generation sequencing in the molecular diagnostics laboratory. During his doctoral studies, he made groundbreaking contributions to the study of hematopoietic stem cells, for which he was awarded the Weintraub International Graduate Student Award and the ProQuest Distinguished Dissertation Award. While a postdoctoral researcher, he made significant contributions to the field of hematopathology, including genomic profiling of lymphoid malignancies, for which he was awarded the Benjamin Castleman Award. So, without further ado, Mark, go on and take it away.
Thank you, Candace, and thank you and welcome to all of our attendees. I'm Mark. I founded Genomic, and I'm currently the Chief Science Officer, as Candace indicated. As a graduate student and a resident and fellow, I saw great promise in the Human Genome Project, which was able to make genome sequencing possible on a wide scale, and then NGS sequencing revolutions making that sequencing for every patient a plausibility. And although I came to bioinformatics and computational techniques a bit late, I was enamored with the power and potential, and not just a little bit jealous of the publication record of some of my colleagues in this field. And I sought to make genome sequencing a practicality by increasing the throughput and capability of genome interpretation using automated techniques. So today I want to do something a little bit different than a standard scientific talk. I'm going to begin by talking a bit about AI in general with a high-level overview for 15 or 20 minutes, and then talk about Genomic's philosophical approach to leveraging advanced computational techniques and machine learning for the purposes of enhancing genome sequencing interpretation, with several specific examples of the approaches that we're taking. So let's begin with a bit of an overview of the state of affairs. I begin with this hype slide, and I begin with a quote from the CEO of Google. He was recently in San Francisco at an investor conference talking about the virtues of AI and its great potential. He's quoted as saying, "AI is probably the most important thing humanity has ever worked on." And he thinks of it as something more profound than electricity or fire. And while it's correct to say that there's great potential for AI, given the exponential ability of AI algorithms to solve problems and the seemingly limitless computational capability at our disposal, these two features of technological advance create a combustible combination with, again, great power and great potential for doing harm, and married that to all of the increasing genomic sequencing data that's available to clinicians and researchers that shows no signs of slowing, there's reason to be optimistic about the potential of AI to solve some of these problems. A separate speaker at a Precision Medicine World Conference in Silicon Valley this year is quoted as saying, "AI will do more for medicine in the next 20 years than all the other biological sciences combined." So I just want to let that land a bit for the audience. It's hard for me to imagine what that would look like. How AI, which almost by necessity will stand on the shoulders of those previous discoveries, could possibly supersede those achievements as opposed to being a simple incremental improvement. So that's the background on the hype and one of the reasons that we're talking about AI in the genomic space. But that's balanced by a number of thought leaders in this space with trepidation. I've called it fear here. This is a quote from no meaner source than Stephen Hawking. There are a number of other leading lights who share his trepidation. But his quote here is, "The development of full artificial intelligence could spell the end of the human race." There are other smart people who know a thing or two about science and scientific history who think that these fears are irrational. And while it's true that there are dangers with any new technology, be it fire or Terminator robots, these thinkers who are throwing a cautionary note are suggesting that AI is far from ready to take over our species. There is, however, a more clear and present danger to the progress of this field that I want to illuminate here in the talk. Many technology vendors, practitioners of AI, are making big promises about their technical capability, and those promises don't often hold up to scrutiny. And many who are witnessing this trend in the lay press are getting the sense of a chilly whisper of AI winters past. So what is an AI winter? It's simply put a decrease in funding for AI research and AI startup companies. And the pattern that brings on these AI winters is simple. Companies or researchers performing AI show early promise that gets hyped in the lay press, leading to a flurry of fantastic outsized investments in a mutually reinforcing cycle. Then there's one of several spectacular public failures that sends the entire system into collapse, with funding and investment cycles freezing over, leading to an AI winter. So let's bring it back down to earth and not talk in hyperbolic terms. Again, this is meant to be a little bit different than a standard scientific talk, so I want to speak at a very high level about what these terms mean that are being bandied about so cavalierly. So first of all, what is AI? AI is simply the creation of intelligent machines that work like humans. There's nothing intrinsic to the definition or the use of AI that goes beyond that. And again, I want to have this land with the audience. I'd like the audience members to think whenever they hear of somebody saying that they use AI, that would be tantamount to a scientist claiming that they are, quote, doing science. Without any qualification or deeper definition or explanation, that's all that AI means, is using machines to perform the work of humans. A more precise definition and a more appropriate term to be used is many of these companies and researchers who are in fact performing AI are using a more explicit technique known as machine learning. So machine learning, put simply, is just math, and these techniques have been around for decades. Machine learning is simply a statistical technique used to progressively improve computer performance without being explicitly programmed to seek out that output. And then another definition of a term that's widely used is deep learning, which is simply a statistical method where computers are used to classify patterns using neural networks. And these networks are referred to as neural because they operate much like human neurons with inputs, transformations, and outputs in an interconnected system. And that system is said to be deep when there are hidden layers beyond the surface at the input level that contain many nodes that perform integrations and calculations on that multitude of connections. Those neural networks can be diagrammed like this. This is machine learning and deep learning represented in schematic form, and I'll walk you through the flow of information from the left to the right, beginning with the inputs, which then is followed by what's known as feature abstraction or identification of salient components of data that are generally superficial. Then there's another layer or many subsequent layers that are hidden of meta feature extraction. And a meta feature you could consider to be a feature of a feature, and so on. Then there's iterative weighting in a comprehensive set of interconnections between those features and the meta features, a summation or quantitation of that information that leads to a defined income, either a yes or a no, or some sort of qualitative dichotomy or a quantitative distribution. That process of defining the weights and the calculus for the feature layers and the meta feature layers can be iterated to achieve a desired outcome for a set of input training data, which is almost always defined by manual or human curation. So an example in a well-studied technique of image recognition would be presenting the computer with the input of an image of a cat or a dog and having the computer dichotomize which images represent cats and which images represent dogs. So in that simple example, features might be ears, nose, eyes, whiskers, etc. Meta features may be the shape of the pupil, the distance between the ears and the nose, an open mouth, a closed mouth, a protruding tongue, or an absent tongue. The weights would be some sort of summation of the combination of those features, where there may be cats who have less than perfectly triangular ears, but nevertheless, the rest of the features and meta features overall tip the balance in favor of the image reflecting a cat. So to put it in less supercilious terms, in any real world example, particularly in genomics, whether you're buying or selling or using AI or doing AI or machine or deep learning, I'd like to have the following questions be kept top of mind. First of all, what explicitly are your inputs? That's a concrete question that deserves a concrete answer. Otherwise, it's difficult to assess the strength of the experimental paradigm that's being used. What explicitly is being identified, and what are the features that are being extracted? Saying that you're going to use electronic medical health record data or EMR data, that's not precisely a feature. Saying that you're going to identify and quantitate drug doses by doing named entity recognition, that is a discrete datum, and that can be understandable to both humans and computers. How is this information being quantitated or assessed? What information is being organized? And then finally, what is the output? How will it be made to be quantitative and useful? Can it be proven that the algorithm is leading to a success? And further, how robust is that algorithm and its output? So the cat and dog example is actually something that image recognition technology is quite good at. But let's get a more nuanced example at play, similar to the nuances that would be faced in, say, clinical medicine when you're trying to segregate patients, make diagnoses, or develop treatment strategies. This is a more challenging permutation of the cat and dog image recognition challenge, which is to say, does this image reflect an image of a dog or a wolf? And this was a challenge that was actually put into operation by a number of AI researchers who asked their algorithm to dichotomize their pictures with training data sets and determine what animal this represented. And a priori, you might expect the algorithm to focus on the feral eyes, the robust mane of this wolf, or the ruddy muzzle, which looks like the wolf had come just from a fresh kill. But what in fact had happened when the AI researchers opened up their AI black box to see what in fact the algorithm was focused on, invariably in pictures like this, the algorithm all but ignored the features of the wolf, which in fact were too similar to the features of the dog, and instead focused on the features of the environment. So it may have been on the surface an effective image recognition experiment, but until the researchers opened up and understood what the algorithm was doing, they had no insight into how that algorithm would behave, say, if it were presented with a picture of a family dog in the backyard during December. So it's a cautionary note that whenever we're doing AI, or more precisely, whenever we're performing machine learning or computational experiments, it's always important to be mindful of what in fact the algorithm is doing. So a very interesting permutation of image recognition technology as it's being applied to genomic sequencing is experiments that were performed by Verily Life Sciences and now involved with Google through Deep Variant, which is to say to examine the quality of next generation DNA sequencing read support or fragment pileups to determine whether the pileups, the read support, represented, quote, a good sequence, one without artifact, or a bad sequence, one with many artifacts that shouldn't be trusted. And so image recognition technology, that existing libraries and paradigms were brought to bear for this relatively straightforward, very discrete problem set with great success. So again, there's great promise associated with using AI to solve some of these problems, particularly now that next generation sequencing is increasingly utilized in the clinic. Larger and larger gene panels, up to and including exome and genome sequencing, are being used for a broader, more diverse patient population. And this data, again, shows no signs of slowing, and the field is struggling to keep up with the required turnaround times when it comes to interpreting this data. But this is in fact a perfect problem for ambitious AI researchers to want to tackle. It's challenging and it has a potentially laudable outcome. But given the volatile mix of big data and AI married to the complexity and importance of genetics and the clinical acuity of the need to get this data interpreted accurately, there's both great potential for harm as well as for good. You may have seen this before. Now that genome sequencing is commoditized, the price points for producing this data are decreasing on a daily basis at a super exponential pace, leading to what has been referred to as the $1,000 genome. The problem that we're trying to solve using AI techniques is rising to the challenge of what's been referred to as the $1 million interpretation, or understanding the next generation sequencing data and helping the human curators and variant interpreters render the interpretation. This has been summarized as the bioinformatic bottleneck, which has not enjoyed the increases in efficiency and decreases in cost like genome sequencing has. So enter into this fray IBM Watson. IBM Watson, as many of you know, has been much maligned in the lay press for having its marketing message supersede its technical capability, with a number of those public failures in their partnerships with a number of different academic and commercial groups. So I want to be careful what I say here. Most of what I'm going to suggest here is written in quotes. And I also don't want to suggest that what IBM Watson has done is without merit. It is simply the case that I was alluding to at the beginning of the talk, that there should be a cautionary note thrown here when IBM Watson is making claims that outstrip its ability. And so we've heard from some users of IBM Watson who have actually seen the output and been part of the training process to produce that output, that there are many benefits to the algorithm and the output that IBM Watson can produce, but overall, I've heard the results summarized as taking clinical advice from a drunk 3-year-old. So a number of terms of art and relevant pieces of information can be assembled using the Watson platform, but overall, to put it euphemistically, Watson's output is not ready for clinical prime time. So this conclusion was adumbrated by a number of practitioners of machine learning and computational intelligence who often would point out that IBM Watson and the claims that were being made about Watson were in fact without merit. That in fact, Watson and IBM were not doing cognitive computing, no matter how many times they said they were. And it was the result of the challenge of needing to collect these seemingly limitless amounts of training data to feed the algorithm so that it could adapt to new inputs, which were invariable every time a new patient was encountered by the algorithm, and its inability to produce interpretable results that extended or let alone matched the results of a standard human clinician. So as a result of a number of these slowdowns and some of the public fallout, a stop or a hold was put on many of these projects, and there has been a compensatory pause among machine learning investigators working in the genomic space. One of those individuals, I think in the context of Watson, is Gary Marcus. I'm quoting here or paraphrasing from his feature article in Wired magazine. This is sort of a crystallization of what the problems with the current state of AI are, as exemplified in the example of IBM Watson. I've summarized this article with an acronym, GOBS, so you can think of how much data was required, the very greediness of the IBM Watson algorithm, requiring as it did huge and thoroughly pre-annotated data sets for it to come up with any sort of semblance of an interpretable result. The results of these algorithms are opaque. They often produce inexplicable output, as I indicated on the previous slide. They're also brittle and crumble with novel inputs, which is another reason reinforcing the need for vast and diversified and thoroughly annotated input training data. And then the results are shallow, and the process itself is shallow because Watson, as it exists, and other machine learning techniques possess no innate knowledge. So this struck me when I was reading through and discussing this article with colleagues. This is exactly the opposite of how humans think and how they approach data interpretation challenges. Humans eschew huge data sets. We don't want them, we can't handle them. Even when we don't understand a problem, what we produce as an output is always understandable given our level of understanding of the input that we're provided. Humans are nimble in their thinking and in fact are expert at adapting to novel inputs with creative approaches and creative solutions to the challenges that are put before them. And the human intellect is deep. We possess our own innate knowledge and our own experiences that have been honed over many decades of living and practicing the art that goes along with the input. And so if AI proper is defined as getting machines to operate more like humans, this is in essence what the failings have been in AI, at least as it pertains to genomic medicine. So pardon the pun here, but to blaze a trail forward, I'd like to propose an alternate approach rather than the GOBS approach that seems to be the only approach being utilized by these large-scale projects. I'd like rather to propose that instead of thinking in a top-down, comprehensive black box approach, I'd like to propose, just as we're doing at Genomic, that a better strategy is to begin with a bottoms-up approach. And I'm going to walk through not only that philosophy that Genomic has invoked, but also provide some specific examples. So the pun here is elementary, my dear Watson. So how are we going to survive this genomic AI winter? There's every reason to be optimistic about the future potential of AI, but it's just as important that while we're maintaining ambitious goals for our applications in genomic medicine, those ambitions should scale linearly with our ability. So a useful mnemonic that at Genomenon we bandy about and focus our efforts on is represented here on this slide, and that's in contrast to the GOBS approach. It's to think small, and this is a play on aspects of health care delivery. When, say, in the context of weight loss, patients are encouraged to think small so that they're sure that they're going to wind up with meaningful results at the end of their weight loss journey. So the first part of the acronym is to be specific. Be specific first, and then have the data extend to additional use cases and new inputs. So a practitioner saying that they're going to, quote, cure cancer is not operating specifically. A better approach would be to focus on specific, clinically well-defined cancer subtypes, and even within that, to focus on specific types of relevant input data, and have that be the ground-up approach as opposed to feeding a black box algorithm all of the data comprising an EMR record for their patient cohort. The next letter is to keep the information measurable. So saying something like our AI will save 100 to 600,000 lives is, to be sure, a quantitative statement with a measured value, but in no way is that a measurable effect. It would be impossible to measure this claim, and having that as a set point or a metric for your own success or the success of your AI project is obviously intractable. The third letter there is to keep the data accessible. So going back to the image recognition wolf example, your machine learning or deep learning algorithms should be interrogable by the researchers and, most importantly, by the clinicians who will be utilizing those results. If the clinicians don't know what their algorithm is doing, who could? And who could entrust the algorithm with informing the patient's care if the provenance of that information is opaque? The fourth letter there is limited. AI algorithms and their output should initially be limited. So this parameter here is different than the specific parameter, just like the breadth of the ocean is different than the ocean's depth. An example or two of how the data should be limited is that you can limit it in terms of time. You can have a focused time period, say post-treatment to some end event, whether it be molecular remission or a demise of a patient. Keeping that limited allows you to have better control over and more likelihood of success for your AI experiments. And then the last one, which is equally important to the other four, is that the output should be likely to be valuable. So your output should be focused on producing something that will be meaningful to clinicians to provide an incremental or more than incremental advance over current knowledge. So an example of something that would not be likely to produce an output that's valuable would be finding that smoking is bad for you by taking data from non-smokers and smokers and understanding a result that was already known to medical science. The question in this circumstance that should be asked here is, what exactly would define success of your AI project, and is it meritorious? Is it worth the price tag and the energy expenditure that would go along with that project? So to bring it down to earth here and put it in the context of what Genomenon is doing with genomic medicine, here are a couple of, quote, small barriers to scaling clinical genomic medicine that Genomenon has isolated and is seeking to solve component by component. So the first is that there's information overload. There's too many papers, too much information in the medical literature describing meaningful, clinically valuable pieces of information that are otherwise difficult to access. That data is highly unstructured, identifying disease gene variant associations from the medical literature, which is one of the components of what Watson is looking to do, is challenging for that very reason. It lacks structure. It requires organization. There's issues associated with variant interpretation. There are, however, the beginnings of an industry standard in the form of codified variant interpretation guidelines that can then be amortized across multiple variants comprising a gene panel, an exome sequencing study, or otherwise a genome interpretation challenge. And then finally, integrating these different data streams in a cohesive and robust way is another one of the challenges to scaling genomic medicine. So put succinctly, the problem that Genomic is addressing is the challenge of extracting and interpreting genetic information, which presently requires a human. And again, to unlock the potential of genomic medicine and promote the ability to scale this practice, to reduce the cost, to decrease the turnaround times, and to make this platform available to an increasing diversity of patients for different indications and in different contexts, solving this problem is requisite. So for those of you who aren't familiar, this is a reflection of the ACMG and AMP, American College of Medical Geneticists and Association for Molecular Pathology guidelines for variant interpretation. So it's a great first effort to make more accurate and more reproducible the process of variant interpretation, which previous to this promulgation of this guideline was an art and required highly specialized experts. It still requires highly specialized experts, but it's beginning to be algorithmatized and codified. So if you're not familiar, or even if you are, there are three major components or data feeds that go into executing this decision algorithm. The first two are very easy and readily available from existing databases. They include population frequency information about individual variants, as well as predictive models of pathogenicity from computational data. So examples of the former would be EXACT and Nomad and ESP 6500, and examples of the latter would be things like Fathom and SIFT and Polyfin and a handful of others. So again, those are fairly easy to access, but they only provide a fraction of the information needed to fully feed and go through this ACMG AMP algorithm. The balance of the information requires extracting knowledge from the medical literature. And that knowledge comes in the form of functional studies, case studies or case series that can give the variant curator insight into segregation data or inheritance patterns or other information about that particular variant or the set of variants that comprise a panel or exome or genome sequencing study for a patient or a patient pedigree or otherwise patient cohorts. That is a hard problem. That is a problem that requires manual interrogation, one that up until Genomic has not been effectively resolved. So Genomic set its sights 3 or 4 years ago on that very challenge, and the fruits of that work come in the form of the Mastermind database. And I say here that Mastermind has indexed the genomic literature, 30 million titles and abstracts indexed on a nightly basis, prioritized according to the content of those titles and abstracts to identify the full text content that is most likely to be contributory to application to those variant classification guidelines. And what Mastermind is looking for when it indexes that reference material is the mention of any disease, any one of thousands in the whole spectrum of human diseases, the mention of any gene, any one of the tens of thousands of genes in the human genome, or any synonym of any of those terms. And then once a gene is identified, to index the rest of that full text information to look for the mention of any one of billions of possible genetic variants, no matter how they may be described by the author, at the cDNA level or at the protein level, using HGVS or conventional nomenclature or some permutation thereof. And then Mastermind has effectively indexed all of that information and then organized and annotated that information, and then has a variety of delivery mechanisms, including the user interface, API calls, and custom database deliverables. This is a reflection of the user interface. It's intended to be a get out of the way user interface where you can input either a disease and or a gene variant, including additional key terms to focus your search and see the information that is most likely to contribute to your specific clinical query. So for the last part of the talk, I'd like to go through a number of the guiding principles that Genomenon uses in every facet of this challenge of organizing and assessing the information from the medical literature. And I've got, I think for each of these slides, I've got a bit of a dichotomy. The first, I would argue the most important, and the first one that we set our sights on, was this challenge of balancing sensitivity and specificity. So put simply, sensitivity is ensuring that you're finding everything that you need or that your users need, and specificity is eliminating those things that you don't, or highlighting the things that are most likely to be relevant to the user's concern. So I've got a couple of iterations of this aspect, sensitivity versus specificity. The first is, where are you getting your information from? So what content is being covered? And I alluded to before, Genomenon's substrate for the information that we're organizing is the peer-reviewed scientific literature, which provides the gold standard of evidence to support claims that are made in clinical circumstances and to drive downstream research or drug discovery. So the content that we're covering, as I suggested on previous slides, is the titles and abstracts of literally every publication in the entire canon of medical literature, and then the full text information that is thoroughly indexed for all of those references that are liberally prioritized, so that we're sure that we're casting a very wide net to put sensitivity first, and then to deal with specificity and refinement to the data that we're showing. We like to consider Mastermind to be a curation tool, and absent near perfect sensitivity, it's hard to be sure that you're not getting all of the information that is required to make a satisfactory and accurate assessment of an individual variant or a set of variants. And to follow along with that, in work that's evolving at Genomenon that we just announced the release of about a week ago now, is we've heard from a number of our users and a number of our clients that increasingly meaningful information is produced and displayed in only the supplemental materials associated with these articles. And so Genomenon has turned its content organization engine around the supplemental information. And already we have many tens of thousands of supplemental references, and that corpus of information is accelerating on a weekly basis. So again, that's a reflection of how Genomenon is putting sensitivity first, and then once we've built that sort of source of truth, comprehensive data set of all the information that we have at our disposal, we are now beginning to work on the specificity of those results. So another aspect of the sensitivity approach, it involves a very technical aspect of doing this indexing work, and that's text conversion. So those of you who are familiar with variant nomenclature recognize that colloquially or following variant nomenclature conventions, there's a number of symbols that are used that can sometimes be rendered in a challenging way in some of the information substrates that we're indexing. So in this case, it's a PDF, And apologies that it's so small, but there's a delta character there that's showcasing the delta F508 variant in CFTR. So that is easily recognizable as a deletion mutation by human variant scientists, but from a computational perspective, that delta character can be rendered as a capital D, which makes it challenging for the indexing process to recognize what in fact that string of characters is supposed to be mentioning or referring to. And so part of our proprietary process is increasing the sensitivity of our search by performing what I've considered microscopic text conversion, which was developed by a very talented software developer on our team internally. And it basically takes a picture of a pixelated map of that character and performs a permutation of optical character recognition on that character to convert it to an appropriate and indexable text form. And in that way, we're able to not miss delta characters or alpha and beta characters that are rendered strangely in different text formats, or more commonly, characters like plus and minus symbols in intronic variants that can be rendered as Thor or M- characters and otherwise perform text indexing, otherwise making text indexing a challenging proposition. To follow on the sensitivity, so there's this aspect of natural language processing or text and data mining called named entity recognition. The example that I'm showing here, there will be three, is diseases and phenotypes. And for this challenge, we're using publicly available open access ontologies for both diseases and in work that's ongoing for phenotypes. So leveraging the work of human curators and data assembly, either from government-sponsored efforts or commercial but still open access information, using that known information as a sort of launching pad for organizing our data set. And I'll emphasize here that our infrastructure is sufficiently modular to include any additional ontology that we have at our disposal, whether it's commercially licensed or also publicly available or bespoke, either developed internally or developed by one of our users. And in work that I'm not going to showcase, there's a number of challenges associated with faithful representations of diseases and phenotypes. A trivial example would be Alzheimer's disease is often abbreviated AD. So too is autosomal dominant, and both of those acronyms can appear in genetic or genomic articles. And so that's a disambiguation challenge that is a very active, ongoing aspect of our research and development team. And having at our disposal this great body of knowledge and the ability to interrogate and refine our algorithms very quickly allows us to make accelerated progress once we've isolated these individual problems. Similar challenges exist for genes, for which we also have an ontology. There's a standard symbol set with aliases, synonyms, and other acronyms or deprecated names coming from the HGNC or Hugo Gene Nomenclature Committee. A couple of challenges to illuminate when doing text and data mining would be the gene HR. It's actually a gene synonym, but you can imagine how HR is often an acronym or an abbreviation for heart rate, and that occasioning a number of false positive matches, which again, in the Mastermind software itself, we show because we put a premium on the sensitivity of the information, but we allow the user to see in context where those terms were mentioned, and can do so also with an automated approach using our own internal infrastructure to maximize the specificity of our results. Other examples would be the gene CES2, which has the alias alpha, which obviously is most frequently mentioned in scientific journals and not referring to the CES2 gene. We have internally ways to identify context cues that indicate that the alpha that's being referred to is in fact not referring to the gene itself, but rather to alpha in a different use. Another challenge is the gene VV, which initially picked up a lot of papers that had to do with varicose veins, which are frequently abbreviated VV. One way that we're solving that challenge internally is by assessing the first mention of the gene name and looking ahead and downstream in the text for a series of words that begin with the letters of that acronym, and identifying those and determining whether those are faithful to the gene symbol or not. And then the last example is a harder challenge to solve. It would be genes that are named either like English words. There is an MICE gene, there is an LARGE or large gene, and there's also a gene that is known simply by the letter T. And so those are some of the challenges that we're facing when organizing this great body of information. And again, our focus is on maximizing sensitivity and then tuning the specificity. Similarly, and in more complicated work that comprises a lot of the proprietary algorithms of Mastermind, is recognizing variants and precisely defining them and associating them with the appropriate genes in that literature. So we've referred to this internally as genomic language processing, recognizing as we do how authors describe the variants, how to interpret those variants at the cDNA or protein level, and how to ensure that we're not missing any pieces of information that are not necessarily faithful to the codified conventional variant nomenclature through HGVS. Another aspect of the computational work that we're doing with the data that we have is favoring pattern recognition over lexical semantics. Lexical semantics is a fancy term that suggests that the computer, quote, understands what is being said. We're seeking to walk before we run, and recognizing the patterns of important information in the literature is certainly a necessary requisite before attempting to make sense of the information as a whole from these references. So a trivial example that I'm including here is authors who publish papers and editors who disseminate those papers know what's important and typically put that important information in display items. So Mastermind in its indexing takes a comprehensive sweep of that full text or supplemental information, including the paragraphs in English language text. But one of the patterns that we're looking for to prioritize the information that's important is the titles and legends of those display items, whether they be figures or tables, because oftentimes there's a summary of the most important information patterned in that information in the literature. Another guiding principle is, rather than filtering out the information, presuming that you understand every time and accurately what a user cares about, is fraught with problems. So instead, Mastermind and our engine at Genomenon seeks to prioritize the information based on what is likely to be important, so that there's no sense of a loss of sensitivity by misinterpreting what the user had suggested they considered important and throwing basically important information out because it didn't match those search parameters. So prioritization is favored over crude filtration, again, in the context of Mastermind being a curation or decision support tool, so that the user can then very quickly make their own conclusion about what is or is not important. Some specific examples of how we prioritize the information. One is that we start at the journal level. We understand through all of the information that we have at our disposal, the titles and abstracts, the full text information, as well as intrinsic knowledge of what journals intend to publish. We can prioritize which journals are meritorious and use that to understand what the user's concern is and prioritize either preclinical, translational, clinical metadata or review information for the user depending on their specifications. Beyond just the journal level, there's the ability that Mastermind has to prioritize the article information, and even more deeply prioritize text chunks or paragraphs or sentences of information that are likely to be the most important component of that reference material, again, based on the user's defined inputs. And then further from that, understanding more holistically which of the variants within a gene, be they associated with a disease or more generically, we recognize which of those variants are most important, or even more broadly in the context of a specific disease, which genes are most contributory to, say, the pathogenesis of a disease, whether it be by virtue of mutations, copy number change, expression level differences, or otherwise gene fusion events. So all of those aspects of the data can be brought to bear to prioritize the information depending on the user interface user's query or the client specification in the context of API deliverables or database assembly projects. A little bit more in depth, there are a couple of aspects that we utilize for prioritization of individual articles. Some aspects that we found to be most important and are included and prioritized in our own prioritization algorithm are the term frequencies in the reference, the gene, the variant, or otherwise key terms. How close together they're mentioned in what's referred to as term propinquity, and where in the reference they're described. If they're described in the title or the abstract, the results section, or the discussion section, or what have you. Term context, additional clinically relevant key terms that appear near to the terms that were searched for, the variant or the gene, depending on what the user has specified, or more generically, whether they're clinically relevant and therefore would prioritize the sentence and then amortize the sentence quality across the entire article to prioritize that article. Another aspect that we prioritize is putting primacy on the evidence. So we like to say we show our work. We don't give ourselves partial credit by not evincing why we thought this variant or this reference or this sentence was important to our user. And that's one of the challenges that groups like IBM Watson has run into, is that it is properly a black box, and the evidence, the path that led up to that prioritization, is not demonstrated. So this is an example of two aspects of how we show our work. We show the term frequencies for each of the references that we've prioritized, and then we go further and actually show the context or the sentences where those terms of interest appeared. Another guiding principle is to begin, again, to think specifically, and then to have those specific pieces of information that have been prioritized and adjudicated be brought to bear more comprehensively to have a general approach to understanding what genes contribute to a disease, say, or what drugs might be meaningful for treatment of a patient with this particular constellation of variants. And then lastly, before I go through a final example, is to do our best or strive to replicate what already has been a human curation process to avoid the problem of the user of that information, who is invariably a human, not understanding the path that was walked to get to that outcome. So again, striving to replicate the human curation process, just doing it faster, more accurately, and more comprehensively. And I'll harken back to the ACMG AMP framework and showcase this screenshot of how Mastermind has coordinated a handful of categorized key terms around those different interpretation criteria, such as in vivo functional studies with all of their attendant relevant keywords. So just to put it all together, I think in the last couple of slides that I have, here's an example of a particular variant that may be encountered in clinical workflow. You can imagine to expand this, a collection of hundreds or thousands of similar variants. This happens to be a missense variant in the UROS gene. URAS is known to be contributory to the pathogenesis of congenital erythropoietic porphyria. But this is a missense variant, and I'm showing some of the population and predictive information. There's no information that suggests that this may be pathogenic by those predictive models. It is known to be a rare variant. And so with those two pieces of the ACMG framework, it's still insufficient information to assess this variant as being pathogenic, likely pathogenic, benign, or a variant of uncertain significance. And so in this example, it was a controlled study that we performed with colleagues of ours at Veritas Genomics. The conventional searches that had previously been performed resulted in 7 references using a complicated multi-parametric search in PubMed, and 120 results using a similar search in Google Scholar. So a great distribution of results. The challenge with the information in PubMed is that it's limited to the titles and abstracts, and with the Google Scholar results, it's crowded with many duplicate pieces of information, many false positives where the gene is mentioned, but the variant is not, or vice versa. And moreover, these results are not clinically prioritized in any meaningful way. So contrast that to the Mastermind result, which returned 66 results that were deduplicated a priori, resulted in no false positive variant gene matches, and were presented in prioritized order with all of the evidence necessary for the user to come to their own conclusions about the strength of that data. And one of the most, I think it was in fact the most prioritized reference, if the functional category in the ACMG schema was included in the search, was this extremely salient Bishop et al. Paper that actually had a very thoroughly characterized cogent mouse model of the disease as a result of the simple knock-in of that missense variant. So if you, as I said, if you amortize this result across all of the references for any one of hundreds or thousands of variants in the context of a sequencing assay, you're starting to see illuminate the vision that Genomenon has of bringing computational techniques to solving the challenge of genome interpretation. So lastly, I think a better question for the title would be not is genomics ready for AI, but is in fact AI ready for genomics? And Genomenon is doing our best to see to it that our bottom-up approach results in success. So with that, I'll turn it over to Candace. We have just a little bit of time to field a couple of questions if there were any from the audience. Let me pass over the control to Candace.
Thanks, Mark. Thanks so much. Wow, a lot of great information, and we got a lot of great questions. So if we were definitely going to run out of time, so what I would like to do is, again, invite people to sign up for Mastermind, to try it for yourself. You can sign up at bit.ly/AI webinar 2018, and this special link will give you a license for the Mastermind free edition, which will start you off with 30 days of Mastermind Professional Edition to try. So what I would like to do is I will take all the questions and send them to Mark. He'll answer them, and we will include them in the web page where we post the webinar. And I just want to thank you again so much for joining us today. I will be sending a follow-up email with a link to the recording and the answers to the questions soon. So be sure to visit us at genomenon.com and follow us on all social media at Genomenon. And if you're interested in using our database in the way Mark demonstrated, you can email us at hello@genomenon.com. Have a wonderful day.